Right. Welcome, Matt Wall, to the Exploring Health and Medicine podcast.
Thank you.
Would you like to introduce yourself to the listeners?
Sure. So my name is Matt Wall. I'm a psychologist and neuroscientist and a little bit of a psychopharmacologist. And I live and work in London. And I do mostly brain imaging with functional MRI and various kinds of drugs like cannabis and psychedelics and sex hormones.
Right. And I'm thinking we're going to focus on classic psychedelics for the conversation today.
Okay.
Would you like to just narrow down for the listeners what are classic psychedelics in contrast to other psychedelics that might not be considered classic?
Sure. So there's a whole bunch of different things that people call psychedelics, but yeah, the classic psychedelics are things like psilocybin from magic mushrooms, LSD, other things like DMT, mescaline, and a couple of others. But they all belong to a particular class that we call classic psychedelics because they all have a similar mode of action. So they all have their main mode of action is at the serotonin 2A receptor or the 5-HT2A receptor. And the other things that people call ketamine or MDMA, psychedelics. Some people say cannabis is a psychedelic, kind of maybe can be. Other things like ibogaine. These things all have somewhat similar effects. Some of the effects are kind of similar, but they all have very different pharmacology. So they all work on very different systems in the brain.
I see. So if I understand you correctly, the classic psychedelics out of all of the substances that you mentioned are the ones that act primarily on the 5-HT2A receptor. For example, psilocybin and LSD.
Exactly, yeah.
Yeah, right. Can you go into a bit more in depth, like what got you into this field and what personal values of yours drive you to engage in this research?
What got me into this field? Well, I first started working on this in, I guess, about 2011 or 2012. So at the time, there was a guy called Professor David Nutt, and he had a young postdoctoral guy working with him called Robin Carhart Harris, and they'd just done the first brain imaging studies with psilocybin that had ever been done, I think. And they were planning a new study using MDMA. And they'd done the previous studies in Cardiff in South Wales. But for this one, they were planning to do it in London, in fact, at the place where I was working. So they asked me to help out with that. So that's how I first started working with the Imperial College Psychedelics Research Group, which they then were. And there was only a few of them at the time. Now there's, I don't know, 20 or 30 people in that research group. And so, yeah, I helped them out with doing the brain imaging stuff on their MDMA study. And then they were planning more studies. They were planning clinical trials for depression with psilocybin therapy. And I ended up working with them on that. And then a second clinical trial for depression. And then various other things have come along. Clinical trials for anorexia. They've done work now looking at psychedelic therapy for chronic pain and for obsessive compulsive disorder. And we're planning studies now in gambling disorder and opiate addiction. So yeah, it's been, I guess, 10 or 12 years. we've been working on this stuff together, and it's been in that time it's really expanded. So 10 years ago there weren't very many people in the world looking at these drugs, but after the first kind of clinical trial results came out, there's been a lot of activity in the area, and a lot of commercial interest, new companies being formed to try and commercialize these things. Interest from other universities in other places have set up psychedelic research labs and things like that. So that's been quite gratifying. And one of the other reasons that I really enjoy working in this area really just came about after seeing the results from the first clinical depression trial, really, which were pretty startling. So for that trial, we recruited treatment-resistant depression patients. So what that means is these are patients that have tried several different treatments before and nothing has worked. So most of the patients that we recruited for that study, they tried lots of different drug treatments. They tried maybe several different kinds of kind of talking therapy. Some of them had even tried more extreme things like electroconvulsive therapy, and nothing had worked for these people. And some of them had been depressed for 20 or even 25 years, with no respite, really. And I'm not a clinical scientist. I'm not a medical doctor or a clinical psychologist or anything like that. So I don't get involved with the therapy on these trials, but I tend to see the patients before and after, because we like to do a scan of their brain at baseline before the therapy, and then usually a few days or maybe a week after they come in and do a second scan. So I see them before and after the therapy. And some of these patients on that first clinical trial were just really different when I saw them again the second time. I saw them a week after, and some of them were just transformed. They looked like different people. And for some of those patients, not all of them, for some of those patients, that brief set of therapy, it was a few days or a week. It was 2 doses spread over a few days or about a week, with some other therapy sessions in between and afterwards. For some of them, it really just transformed them. And these were patients, like I said, where nothing else had worked. And that's when I first started thinking, maybe there's really something to this. Maybe this is really something we need to focus on and explore a bit more.
Yeah, I could definitely see how seeing such a transformation of the patients that you met would induce an interest of exploring the effects of these psychedelic substances more in depth. If you look at psilocybin, which is, to my understanding, the classic psychedelic substance that has been most studied in clinical depression. If you look at its effectiveness in clinical depression, I guess it may depend on whether the studied group is treatment resistant or not, but you can take the question and answer it as you like. But how effective on a population level, so to speak, is psilocybin for clinical depression?
It's a little hard to say, because most of the studies that we have are still quite small, unfortunately. And the treatment resistant thing is a bit difficult as well. So we've got some studies where they have recruited treatment resistant patients, and other studies where they've just recruited more kind of standard depression patients. But I mean, what seems to be, as a general rule, roughly speaking, it seems to be about a third of subjects have a really good response to the treatment, and they show this immediate antidepressant effect, and that is maintained for some weeks or months. So it kind of varies across studies how long people tend to follow up. So some studies do 3-month follow-ups, some studies do 6 months. There's at least one study that's followed people up for a year and find that quite a few people seem to have enduring effects for a year. So within the confines of the study, whether it's a 3-month or a 6-month follow-up, you tend to have about a third of patients that you can say, okay, they're cured, right? We've cured them with this one treatment or a couple of treatments. Then you get about another third of patients where they have a really good response initially, but then over the 3 months or the 6 months, they tend to, the effect kind of fades and they tend to just drift down into depression again. And then you have the final third of patients where it just doesn't seem to work as well for them, and they may show a little bit of response initially, but not to the same extent as the others. So it's kind of, that's how it seems to divide in most of the studies. So it doesn't help everybody. And one of the key questions that I think's around at the moment is, well, what's different about these 3 groups, and can we change things around a little bit to maybe, okay, we've got the one group where it works really well, perfect, let's not touch those. We've got the middle group where it works pretty well at first, but then the effects fade. Well, what can we do with that? Can we think about changing the treatment protocol a bit, giving them some more kind of follow-up therapy sessions to try and maintain the effect a bit more, maybe even think about additional kind of booster sessions of drug dosing every 3 months or something like that. Can we do more to help that group? And then we'd really like to know about the final third as well, the people that it just doesn't work very well for. What's different about this group? And these are all really important questions, but like I said, because the all the studies that we have are still relatively small, like the 2 Imperial studies, we're talking about 20, 25 people. Once you start breaking those down into even smaller groups, you end up with 7 or 8 or 9 people in each group. You just don't have the numbers to be able to do anything reliable. Ideally we'd love to be able to look at their data from their brain scans and say, okay, this group are different from this group, are different from this group in some way. But with the kind of numbers that we have at the moment, it's just not really, it's really hard to see anything reliable like that.
Yeah, I understand that. Do you think that will be, like, are there any such studies on the horizon currently?
Yeah, I mean, they're starting to do bigger studies. I mean, there are much bigger clinical trials that have been done. So there are clinical trials with maybe a couple of 100 patients, which are happening, which is great. But the problem is with those, they don't tend to include the brain scanning part, right? Because the brain scanning part is really expensive. And if you just want to show that the drug works and helps people, and if that's your priority and you've got limited resources, which everybody has, then you probably don't want to scan 200 people's brains as part of your clinical trial, as well as putting them through the dosing. So the brain scanning stuff tends to happen on a smaller scale, which is a bit unfortunate, because then we're always dealing with small numbers. But as time goes on and we get more more studies happening, and hopefully a bit more funding and resources available, then I hope we're going to get some more and better quality data on that. And once we build up a few studies as well, I mean, there's a movement at the moment in the field to start sharing data as well. So you can, hopefully we can start kind of pooling data from all these smaller studies and start saying something a bit more interesting about that kind of thing as well.
Yeah, yeah. Yeah, I've been thinking about ways to, like, for science in general, if, like, for example, the internet could be leveraged in some way, like if data could be gathered in a reliable way through the internet. So I could see how that would be applicable here as well.
Yeah, well, I mean, there are lots of I mean, the kind of areas that I work in, which is, you know, brain imaging, there are lots of initiatives like that that are aiming to kind of facilitate data sharing across institutions and internationally and so on. We haven't got one specifically for psychedelic brain data yet, I think, but I think it's coming. I've definitely heard people talking about that kind of thing.
Yeah, yeah. All right. You mentioned that there were some larger studies on, if I understood you correctly, psilocybin for depression, that don't have brain imaging data, but that do have depressive symptoms data. What do those studies say about the effects of psilocybin on symptoms of clinical depression in clinically depressed patients?
So I think the biggest study that we have now is a study by a company called Compass Pathways, and I think they had about 200 people split into 3 groups. So that what they did was something quite clever, which was that they used 3 different doses of psilocybin. So they used 1 milligram, which is a really tiny dose. It's like inactive, almost a placebo, basically. 10 milligrams and 25 milligrams. So I think they had about 75 people in each group. And what they found was that the 1 milligram didn't really do anything. The 10 milligram, people had a reasonably good response initially, but the effects didn't really last very long. And after a few days or weeks, it was quite similar to the placebo or the inactive dose, the 1 milligram. But the 25 milligram group did show a strong initial effect, and that effect was sustained up to about 3 months, I think. So what was nice about that is that you had this dose response effect. Because one of the big critiques about psychedelic clinical trials is that you can't adequately control them. You can't really have a placebo condition. I mean, you can do it, but you can't maintain the blinding of the trial, because everybody knows when they've been dosed with a 25 milligram dose of psilocybin, say, because you have this very strong subjective effect. So that kind of necessarily unblinds the trial, both for the patient and for whoever's doing administering the doses. But with that kind of design, where you had these 3 levels of doses, I think that does help to get around that problem a little bit. If you can show that a 10 milligram dose is not as effective as a 25 milligram dose, I think that does help to show that it is really the drug doing something rather than just a placebo effect.
I understand you correctly, it's very difficult to have a true placebo measure in psychedelic science, but it is possible to use other ways to strengthen the evidence as regards whether there is a causal effect of the psychedelic substance?
Yeah, I mean, there are things you can do. So you can use this multi-dose design I've been talking about. You can have active placebos, so you can have other drugs which will give you some kind of subjective effects, which are not psychedelics. So there's a recent paper where they compared psilocybin. This was healthy controls, and it was kind of a more of a neuroscience paper than a clinical trial. But they compared psilocybin with methylphenidate, which is a pretty standard kind of stimulant, just to compare the brain effects. So you can do things like that, and that does help to. It doesn't completely get around the problem, but it helps to kind of obscure the drug dosing, I think. So people may not know or might not be sure that they were getting or which drug they were getting.
Yeah.
But it is a problem. But it's a problem. It's one of the things that people who are critical of psychedelics like to really point to. But it's a problem in a lot of different drug trials, even like kind of standard depression treatments, things like SSRIs, selective serotonin reuptake inhibitors. People very often tend to know when they're getting the real drug or a placebo, even in those trials, because they have effects, right? Whether they're side effects or clinical effects, these drugs do have effects on people. So it's not something that's unique to psychedelics at all.
Yeah. As regards the comparison between psychedelics and SSRIs, other than psychedelics acting very quickly and having a sustained effect for weeks to months after the dosing session, are there any other pros or cons of psychedelics in comparison?
Possibly compared to SSRIs. So,
SSRIs.
I mean, one of the Imperial studies that I worked on actually did compare psilocybin with a drug called escitalopram, which is a standard SSRI. And what we found in that study was that both groups showed actually a pretty similar antidepressant effect. So we had a 6-week kind of trial protocol, and the one group took escitalopram for 6 weeks, and another group took a placebo pill for those 6 weeks and had a couple of psychedelic dosing sessions in that time as well. And what we found really was that over the course of the 6 weeks, the antidepressant effects were actually quite similar. So maybe a little bit superior for psilocybin, but not really hugely significantly. But there were other measures. So we used a bunch of other measures, including measures of well-being. So there's a scale called the Warwick-Edinburgh Mental Well-being Scale, and psilocybin was definitely better on that scale. So that focuses on, rather than the depression clinical scales tend to focus on kind of negative things and how bad you're feeling. The Warwick-Edinburgh Mental Wellbeing Scale tends to focus on positive things like how well you're doing in your life, things like that. And psilocybin was definitely better on that. And in fact, psilocybin was better on pretty much every other clinical scale that we looked at. So yeah, there are some advantages, but again, these are all different tools in the toolbox, if you like, and what works well for one patient might not work well for another. You know, one patient might do a lot better on SSRIs, and another patient might do better with psychedelic therapy. And And at the moment, it's kind of hard to say for any particular patient what that might be.
Yeah, I see. There's just not enough data to say. Yeah.
And we don't know what to look at really, even. You know, is it genetic differences? Is it differences in gene expression in the brain? Is it something to do with a history of childhood trauma that's contributing to gene expression? Is it you know, with kind of epigenetic changes? Is it some aspect of brain function? Is it some way that different way that people are metabolizing these things? Or is it, you know, possibly all of the above? You know, it's really difficult. And you can do studies where you can, you know, compare, say, a group of depressed patients and a group of healthy patients, healthy people, sorry. And you can scan everybody's brain and you can compare this group of 50 depressed people and 50 healthy people, and maybe you can find some difference in their brain activity, for instance. That's fine, and that's interesting and scientifically worthwhile doing, but you're still looking at a set of two groups of people, and if you plotted their data together, the distributions would just overlap completely, and there'd be a small difference between the groups. So for any one patient within those groups, they're probably going to be somewhere in the middle of those two overlapping distributions. And what that means is trying to develop a good diagnostic biomarker for depression. We can't just scan one person's brain and look at, say, the hippocampus and go, oh yeah, you have a broken bit of brain in there and that's why you're depressed, because they're always going to be in the middle of these two sets of distributions somewhere. We haven't found really, and people have tried, we haven't really found a good, useful biomarker that's derived from brain imaging technology, which is actually helpful in psychiatric disorders like depression. It's not like if you go to your doctor and they give you a blood test and there are certain protein markers in your blood, then your doctor will know that you have cancer, for instance. And it's very clear, if you have these proteins in your blood, then there's a tumor somewhere in your body that's producing them, and you definitely have cancer. There's nothing like that for psychiatry. You can't look at any kind of biological or functional marker and say this is why you have depression or schizophrenia or ADHD or whatever. And this is going to be, I think, a really big focus of research in the next decades, is going to be really trying to drill down and figure out exactly why that is and whether we can actually derive anything useful from these technologies in that respect.
Right. And this type of research is the type of research that you have been taking part in conducting, right?
Well, we're starting to. So one of the things that would be great to figure out is, as well as, being able to diagnose psychiatric disorders like that, it would be great to know what treatments people are going to respond to. So it'd be great to know if we could scan somebody's brain and then we could say, okay, this treatment will work for you and this treatment will likely work for you. Because what happens, particularly with depression, but with a lot of other psychiatric disorders, is that patients will cycle through quite a few treatments before they actually find one that works. So they'll try one drug, they won't like that, it gives them nasty side effects, it doesn't cure their depression. They'll try a different drug, then their doctor will say, okay, let's try a combination, let's try a little bit of this and a little bit of that, and then eventually hopefully they'll find something that works. But that process takes, I don't know, 2 or 3 years maybe, and all the time they're going back to their doctor, and their doctor's trying to find things that'll work for them. So if we can in some way figure out in advance what treatments are more likely to work for a particular patient, that will be a huge benefit. And it doesn't have to be a perfect answer. If we can cut that time where they're cycling through treatments, if we can cut that from 2 years to 1 year, and they only have to try 2 different treatments before they find the right one, then that will be a huge benefit for the patient. We don't have to get the perfect answer, we just have to improve things a bit. So I think there is quite a lot of scope there, hopefully, for doing this kind of work and trying to figure these things out, and hopefully making a good generating some good benefits for patients.
Yeah, I could definitely see how that would be very useful and beneficial for patients if the fMRI, if they could just go into the fMRI and then the doctors could help them within 1 year based on that data rather than 2 or 3 years. So I could definitely see the value. Before we dive into the effects of psychedelics as measured by primarily fMRI in your research, can you describe some key terms as regards the methodology or what is actually measured rather in your fMRI research, such as arterial spin labeling, cerebral blood flow, and RSFC as well?
Oh, RSFC, yeah, okay.
Yeah.
Sure. So so fMRI, functional magnetic resonance imaging. So it's an adaptation of normal MRI. So normal MRI, if anybody's ever been in an MRI scanner in a hospital, you've probably had an anatomical MRI, which is using a scanner to basically take pictures of the inside of the body, and that's really useful clinically. In research, we often tend to rely on something called fMRI, which is a kind of adaptation of MRI, which is focused at looking at changes in oxygen concentrations, basically, in the brain tissue. So when an area of the brain becomes active, it needs more oxygen and more glucose from the blood in order to remain active. So the vascular system in the brain is quite reactive, and when an area becomes active, it will dump additional oxygen in that area. And because oxygen is carried in the blood by hemoglobin, which quite handily is quite magnetic, because the hemoglobin molecule has an iron atom at the center, which makes the hemoglobin molecule quite magnetic. We can see that local change in oxygenation as a change in the magnetic signal that we get in the scanner. So what that means is we can, rather than taking just a static picture of the brain, we can instead take something that's a bit more like a movie. We can take, you know, a few hundred frames over the course of, 10-15 minutes, and we can see changes in the concentration of oxygen at different parts of the brain. Now, you can do that in broadly 2 ways. You can do it with a task, so you can get people to look at pictures, watch videos, listen to music, do little cognitive tasks, play games where they try and win money, or where they complete puzzles and things like that. So you can actually get their brain kind of working in particular ways, and you can see the effects of that task or those stimuli. Or you can do something called resting state fMRI, which is where you ask people to just relax and don't do anything, and you scan their brain, and you get a kind of baseline readout of brain activity when you do that, because the brain is pretty active all the time, really. Even when you're lying still and not really doing anything, bits of your brain that talk to each other all the time are still talking to each other. And those bits form what we call resting state networks. So there are a set of networks in the brain. The brain is not a homogeneous lump. It's divided up into lots of different regions that do different things. And the regions all talk to each other and form these particular networks, which underlie different functions. Did that cover anything? So yeah, RSFC is resting state functional connectivity. Which is what you see when you, or one of the analyses you can do when you do resting state fMRI, and you're looking at the connection between different brain networks.
Right. So
Did that
can
cover
you
everything?
describe? Yeah, it was a great description of fMRI, I think. Can you go one level deeper on RSFC, like the functional connectivity part?
Yeah.
Is it measuring activity then in a certain brain network, a certain connection pathway in the brain?
So you're looking at, what you're really looking at is correlations. Okay, so you're looking at regions of the brain which are doing similar things. So if you have two regions of the brain which are kind of spatially different, so one at the back and one at the front, say, but when you look at what that area is doing over your, say, 10-minute scan, if the time series of that activity is similar in those two regions, so if when it goes up in one region, it also goes up in the other region, and when it goes down in both.
Yeah.
The implication is that those two regions are what we call functionally connected, meaning that they are exchanging information. There must be some communication happening between those two regions in order that they are synchronizing their activity.
Yes, and that can be indirect as well. Like
it could be, yeah.
Two areas aren't anatomically connected, but still functionally are.
It could be indirect, yeah. I mean, there are limitations to that assumption, right? So, it could be a third area is influencing both of those two areas kind of separately, if you like.
Yeah.
But the general assumption is that if two areas are doing similar things at similar times, then they must be communicating in some way.
Yes. Yeah, it was a great description of like a very understandable description, I think, of fMRI, and how it's used in the psychedelic research field as well. As regards arterial spin labeling, I'm not sure I understand what that is still.
That's so that's actually a different kind of technique. So that is it's a kind of still a kind of functional MRI, but it's not a dynamic technique like the fMRI I was just talking about. So rather than taking a movie, you get a static image, a bit like an anatomical MRI, but it's an image of cerebral blood flow. So you do a scan and you get a kind of a snapshot of the blood flow in the brain, or how much blood is flowing into and out of the brain at any particular time.
I see. Yeah. Okay. So let's go into the effects of psychedelics on the brain, and let's start with like from a zoomed out perspective. So I'm thinking about global network integration, and also on a general level of the modular integration or disintegration. Do you want to explain the effects of classic psychedelics on
Sure.
global?
So what we've been finding, and again, I have to emphasize, these are all relatively small studies, but this is probably the most robust finding that we've got from all these different relatively small studies. What we find is that classic psychedelics have a pretty unique effect on the brain in that they tend to randomize the connectivity patterns that we see. So in the normal situation, you have all of these brain resting state networks talking to each other and maintaining their communication even when you're doing nothing. So like, even if you've got, so all of the visual bits of the brain at the back of the head are all quite tightly integrated and talk to each other and maintain their communication even when you have your eyes closed. All of the motor bits of the brain at the top that are responsible for your motor actions, even when you're not moving, they're still communicating and coordinating their interactions. What happens when you give, oh, and one other thing, they're all of these networks operate as fairly independent modules. So the visual bits of the brain are all talking to each other, and the motor bits of the brain are all talking to each other, but there's not much connection between the motor bits and the visual bits, right? You can think of the brain as a set of reasonably independent modules that all function as independent modules, but also all kind of integrate together. What happens when you give psychedelics is that that modularity of the brain goes down. Okay, so you have less activity within networks and you have more activity between networks. So that whole kind of coherent structure that we see in these resting state networks with these independent modules, it breaks down quite profoundly. So you get much more random connections between areas that don't normally talk to each other, and you get weaker connections between areas that do normally talk to each other. So psychedelics have the effect of almost of like randomizing brain activity, and that's why we think they have such strong subjective effects on, you know, perception and consciousness and our experience of the world and things like that.
Yeah. And now we're talking about the acute effects during the dosing session, right?
Yes, exactly. Yeah.
Yes.
Yeah.
And how are modular and global levels of network integration affected by psychedelics in the long term, like on a month perspective?
Wow, that's a good question. So yeah, the effects do persist for a while at least. So there hasn't been many, in fact, there's only been one study which has actually looked at the acute effects and the long-term effects in the same group of patients. But what they've found is that the acute effects persist for about 3 weeks. So for about 3 weeks after a dose, you can still see changes in network integration and modularity and these kinds of measures. After about 3 or 4 weeks, it kind of returns to baseline. So what that suggests is that there's a window there, and the way that we think that psychedelics have their clinical effects is related to this. So there's a window of opportunity and a window where the brain is put into quite a flexible and plastic state, and it's a bit like almost returning the brain to a state when you were a child. So if you look at resting state fMRI studies with children, you find a similar kind of features to psychedelic drugs. So the the pattern of connectivity in the brain is much more random. And the reason for that is that the adult resting state networks haven't formed yet, right? So during development, child's brains are much more flexible and plastic, partly because their brain hasn't settled down into the adult patterns of networks yet. So, I mean, as a kind of analogy, you can say that psychedelics put the brain back into almost a childlike state for a fairly brief window of, you know, days or weeks and so on. And we think that what that enables is people to, with the right therapy and the right kind of psychological support, it allows people to break out of the thinking patterns that they've been dominated by, perhaps with depression or addiction or obsessive compulsive disorder or eating disorders and things like that. Allows people to get out of these dysfunctional behaviors that have become very kind of trained and ingrained, and it allows them to perhaps weaken those relationships and develop new, more functional, more healthier, better ways of thinking about their disorder and better ways of behaving. And that's perhaps how psychedelics have their positive clinical effects. And the neat thing about that is it's not really tied to any particular disorder. So potentially, if that is how they work, potentially it could be used for almost any disorder where people have very stereotyped or ingrained patterns of thinking and behavior. So things like depression, addiction, post-traumatic stress disorder, eating disorders, obsessive compulsive disorder, chronic pain. It could potentially help almost any of these people if that explanation is correct.
Yes, yes. I think, and that is, of course, a very, like, that would be very, very good if it was the case. But I think that there are also some fears that have been more or less prevalent in the collective unconsciousness, or in the culture, so to speak.
Yeah.
One of which might be that, okay, so since psychedelics have this brain rewiring effect, mightn't there be a risk for losing skills or having a negative effect on cognitive function, for example?
Well, I mean, I think that's a serious, I think that's a possibility that we have to consider seriously. Yes, because if you are putting the brain into a very flexible and plastic state, that's an entirely kind of neutral process, if you want. So yeah, there is the possibility that you could then develop different but equally negative ways of thinking and behaving. Sure. And I think this is why people, most people anyway, are very keen on the idea that these drugs should only really be used with appropriate therapists and guides. So it's, you know, these are not just drug treatments. At least that tends to be how most people think about them. It is psychedelic assisted psychotherapy.
Right.
I mean, some people just call it psychedelic therapy to cover everything. But yeah, I think most people in the community are pretty clear on the idea that you need to use these drugs with appropriately trained people, with the right therapists for whatever condition you're dealing with, and you need to have really good preparation sessions beforehand to let people know what's going to happen, the kind of things that they're going to feel. You need to have good support while people are actually being dosed, and then you need to have possibly quite extensive follow-up therapy sessions, and people call them integration sessions, where you talk about the experience and you try and think about how to integrate some of the insights into your life and that kind of thing. So yeah, I think that's probably super important, and it's not going to be the same. Doing it like that is not going to be the same as people just using these things, recreationally, which happens a lot. And there is, with recreational use or kind of other kinds of like non-clinical use, I guess there is the opportunity of negative effects, sure, because these are powerful drugs, right? These are the most powerful drugs that we know of, in terms of how they change our consciousness and our perceptions and things like that. So yeah, they are, sharp blades, if you like. You can do a lot of good in medicine with a sharp blade, but you can do a lot of damage as well, right?
Yeah, of course. Yeah. So if I understand correctly, the effects of psychedelics are present and potently so during the acute dosing session in the brain. The global network activity integration is increased during the dosing session, and the modular activity is decreased or disintegrated.
Exactly.
And the brain effects correlates to psychological effects during the acute dosing session, where these psychological effects are of an increased level of openness, right? Where there is a possibility for good or bad conditioning. So there's a risk for negative long-term effects, and there's also a potential for therapeutic effects.
Yes, I think that's probably true. I mean, I don't think there's a lot of evidence that people end up with negative long-term effects. So I mean, even the patients that have participated in our trials that have had what tends to be called a challenging experience during the dosing sessions. So a lot of people, people used to call it a bad trip, right? The biggest adverse event that we get is anxiety, right? And a lot of people, particularly when the effects first start being felt and they get that initial feeling, they get quite anxious. For most people, they then calm down and it's fine. Other people have, some more extreme emotional reactions. But even those people that have maybe a bit of a hard time during the dosing sessions itself, don't really report any negative long-term effects. And that also doesn't seem to be related to the clinical effects. So you get some people that maybe had a really difficult time in the dosing session, but then still show really good antidepressant effects. But yeah, I think there is the potential there for possibly negative effects to creep in during that window of opportunity. But I mean, I think it's something that everybody who's running studies at the moment in this is fairly careful about, and to provide good psychological support before, during, and after, to kind of guard against that. There's some amazing work from an animal preclinical researcher in the US called Gal Dolen. So I was talking about how psychedelics return the brain to a kind of almost childlike state. And what she's shown with mice and rats is that psychedelics reopens critical periods for learning. So, you know, things like learning languages. Learning languages is very, very easy when you're a child. You just do it naturally. You just pick it up if you're exposed to the right language at the right time. Learning a new language when you're an adult is much harder, right? So there's this critical period for language learning, and there are critical periods for various other kinds of learning as well. What she's found is that if you get a bunch of rats to run a maze, the young ones will learn it faster than the old rats. But then you give the old rats a dose of psychedelics, and then the day after they will learn a new maze just as fast as the young ones. So her whole interpretation of this is that psychedelics reopen these critical windows for learning certain things, which is quite an exciting prospect.
Yeah, definitely. Do you think that this could mean that they might have some use in developmental psychiatric disorders as well?
Oh, that's a good question. So, I mean, I have seen suggestions that they might be useful in things like autism, ADHD, other developmental disorders. It's a pretty big kind of conceptual and ethical hurdle to get across, giving these kinds of treatments to children, I think.
Right. Yeah.
Even disorders like eating disorders that tends to come on for at least some people in maybe, you know, early teenage years. I think even that might be a stretch. There are so many, you know, in most places of the world, these drugs are super illegal drugs, right? Like here where I am in the UK, the same in the US, they're what's called Schedule 1 drugs, which means legally they are they carry the kind of maximum penalties for drug possession, and they're recognized, or they're legally classed as having no medical use, right? So we've got a lot of kind of legal and ethical concerns to get over with these drugs. So I'm no expert on developmental disorders at all, but I have seen some, you know, chatter about possibly wanting to use psychedelics for things like that. But I think we're not there yet. I mean, I think we've got to try and get these treatments licensed just for simple things like depression and PTSD, and take it from there, really, I think.
Yeah. I could definitely see why it would be difficult, and also ethically a more difficult question whether it's good to try these substances in children.
I think we'd have to have some really good. I think we'd have to have some really good preclinical data, and just some really good hypotheses about how exactly these things might be able to help whatever neurodevelopmental disorder we're talking about, you know. I don't think we're there yet.
Yeah. I mean, there's always this question when new treatments are in question of what the long-term effects and possible unknown side effects are, right? And
Exactly,
even more
yeah.
so in children. So
Exactly. Yeah. You have to be really sure about any kind of drug treatment for kids, I think.
yeah, I agree. Okay, so we were talking about modular disintegration during the acute dosing session. Is it so that it is continuously disintegrated for a few months after the dosing session, or is it, is it that way, or?
I mean, it's hard to say, right? So, I mean, we've got quite limited data. Like I said, there was a study last year where they scanned people in the acute state and then did various follow-up scans afterwards. And they showed an effect that was detectable up to about 3 weeks, I think. But, you know, whether there are effects that persist for longer than that we just can't detect because we're looking at well, because maybe our tools are insensitive, or we're looking at the wrong aspect of brain function. I don't know. So there's also this idea that psychedelics increase neuroplasticity. Now, neuroplasticity is a very broad term, right? So you can think of these changes in brain function that we've been talking about. You can think of those as a kind of neuroplasticity effect because it changes over a period of days or weeks. But there's really good data showing, just from studies where they've got cultured neurons in a petri dish in a lab, showing that psychedelics can bind to certain receptors in the brain which are related to neurotrophic factors, like brain-derived neurotrophic factor, which basically grows brain cells. And there are studies looking at number of synapses on a neuron, and you dose the neuron with psychedelics, and you find after a few days or a week or something that the neuron has grown new synapses. And psychedelics seem to be quite powerful at that. So there are lots of drugs that do affect neurotrophic factors and have similar kinds of effects, like SSRIs, or cocaine even has positive effects on neuroplasticity. But psychedelics seem to be a particularly powerful way of doing that. Now, there's only a tiny little bit of data which suggests that these effects are present in humans. We think they probably are, because they're present in rats and mice and animals, where we can basically cut their brains up and see these effects. With humans, it's a bit more ethically challenging to cut their brains up, unfortunately. So we have to resort to these things that I use, which is, non-invasively looking at their brains with MRI scanners and things like that. So there's only really a tiny bit of data in humans. We're actually doing some studies where I work now with ketamine and DMT, which are using fMRI plus another imaging technique called PET, to try and look at changes in synaptic density with psychedelics. And we don't really know how these neuroplasticity effects might be related to the longer term effects. It might all just be the same kind of thing. Okay, we're putting the brain into this very plastic state. We're allowing new synapses to form and new connections to form. But that's a very kind of broad way of looking at it. Neuroplasticity can mean everything from the level of an individual receptor or a synapse in the brain up to these kind of large scale functional connectivity changes that we've been talking about, changes in modularity of whole brain networks, and there's a lot of different levels in between that, where things might be changing as well. So we've got an insight into maybe some of them, but the process, we don't know all the processes that are going on, and we don't know how any of them really relate to the clinical effects.
I see. So it's still a field that requires a lot more research to be at a state where it has its certainties as regards, like, okay, this is definitely what is happening here, and yeah, and so on.
Yeah,
I see. Okay.
But we still don't really know how caffeine works in the brain. Okay.
Yeah.
Caffeine, the most common drug in the world, right? That a big proportion of people in the world use every day. We're still trying to figure out how caffeine works. So, yeah.
Yeah.
Still lots to do.
Yeah, that's true. But still, people. I feel like people are more open to caffeine than they are to psychedelics, even in the context of a medical treatment or a psychedelic assisted therapy.
Sure. Well, caffeine's a lot less scary and weird than psychedelics, for sure. Yeah.
Yeah, it's less psychoactive and potent in that sense. Yeah.
Sure.
Okay, so going back to the effects of classic psychedelics on the brain, can you go into how they affect the default mode network?
Sure. So, I mean, there is a quite a strong kind of narrative in the research around the default mode network, but I think what we're finding really is that psychedelics pretty much affect all brain networks, and the default mode network is one of the major brain networks, and it really serves the role of, I think, coordinating a lot of others. So the default mode network is most active when you're doing nothing. So when you're sitting or lying quietly and kind of focusing internally rather than focusing on any external task, your default mode network is most active. And people talk about the default mode network therefore representing something related to our phenomenal consciousness or our experience of being a kind of conscious agent. It's probably not as simple as that. Like, the default mode is not consciousness, for sure, but it's probably something related to that. So because I think it's the most prominent and obvious network in the human brain, like you can take pretty much anybody's brain scan data and you run it through a certain type of analysis and you can see their default mode network pops out, right? It's one of the clearest and most obvious. And I think because psychedelics basically affect the whole brain, they probably affect the default mode network more, just because it's the most, it's one of the strongest and most prominent networks. And it does have the role of, I think, kind of coordinating activity in lots of other networks. So knocking out the default mode network might have these kind of widespread effects that we see with psychedelics. But I think really we see a more kind of general effect of psychedelics. I don't think it's just related to the default mode network, for instance.
Okay, I see. So I was thinking that let's take a cut here and just talk about the rest of the conversation.
Sure.
And I'll cut this out. So I have a long list of brain areas that I went through several of the studies you have co-authored, and so I have this long list of brain
Did I
areas.
just ruin your plan? Sorry.
Yeah, kind of.
Yeah.
But it's very good that you say that and make that clear, because
Right.
the important thing is to get the essence across to the listener, right?
Mm-hmm.
But so I'm thinking maybe we can focus in on some of these brain areas. Are there any specific brain areas that you think are particularly interesting as regards the effects of psilocybin or LSD on these brain areas?
So yeah, so there are brain areas in particular that people have shown to be affected by classic psychedelics. So I mean, one of the things that we've done in our clinical trial research with depressive patients is to look at the amygdala, and the amygdala is really a key area involved in emotional responses. And it's quite a common thing in depression research when people are looking at standard antidepressants. So they do this task where they show people emotional faces on the screen. And what you find when you show people emotional faces is that their amygdala responds, because it's one of the key areas involved in responding to emotion. And when you give people a standard antidepressant treatment like SSRIs, what you often see is a decrease in amygdala response, particularly to the negative emotions. And the interpretation of this is that SSRIs are reducing the response to negative emotions, and therefore reducing the kind of bias that depressive people have about the world, which is that they tend to think that the world is very negative. And when they look at the world, they tend to see the negative side of things. And what we found in our data is that in the first clinical trial, psilocybin actually increases amygdala responses to emotional faces. And in the second clinical trial, where we compared escitalopram with psilocybin, we see the standard effect with escitalopram, so we get this reduction in amygdala responses, and we see no change really in the psilocybin therapy, even though we see good antidepressant effects in both these groups. We see this effect, this change in response to emotional faces in the escitalopram group, where it tends to go down, and in the psilocybin group we see no change. So that really suggests that there's quite a different mechanism working there. We've got antidepressant effects in both groups, but we've got this effect on emotional responsiveness, particularly in the amygdala with the SSRI group. And it might be related to some changes in emotional function that people often report with SSRIs. So people often report this emotional blunting effect, where they feel like the depression has lifted a bit, but they also feel like they don't respond to other emotional situations in the same way. So they don't cry at a sad movie anymore, or they don't enjoy listening to music quite as much as they used to, or things like that. So people often report this kind of general effect where it feels like the drug has kind of put a blanket over everything. So that's something that we pursued, particularly with the amygdala. In our other studies where we've got music stimuli, we've shown effects in areas that are related to positive emotional effects of the music as well. So areas like the parts of the reward system, which is the ventral striatum, areas in the middle of the brain, which are very dopamine rich and are quite involved in the emotional response to music. So we see changes related to psychedelic therapy there. All of these things, again, I keep saying this, we've got limited data and we've got limited tasks that we've used. So, you know, the kind of brain areas that you can talk about depend on which brain areas you target with particular tasks. So, you know, if we are interested in the effects of psychedelics on the brain's reward system, maybe we'd use a task where we get patients to play a little game and win money, right? And what you find is that when patients win money when they're playing this game, you get a little burst of activity in their reward system, and their reward system lights up. And there probably are effects of psychedelics on these systems, but nobody's done that study yet, right? Nobody's looked at the reward system with psychedelics. If you're interested in, you know, purely kind of cognitive effects on memory and things like that, you design a task which is designed to hit those brain areas, and then you look at how it changes with psychedelics. Nobody's done that yet either.
Yeah, I see. Yeah, but so did I understand you correctly that there have been no studies on the limbic parts of the striatum?
Not that I know of that have used particular tasks that are designed to activate those areas. So we've actually, some colleagues of mine, my friend and colleague Natalie Ertl has just put out a little paper, or a preprint actually, where we actually have looked at striatum resting state connectivity. So pretty much all of the studies so far that have looked at psychedelics have tended to just use the cortex, the outer bit of the brain, the grey wrinkly bit that, you know, that looks like a brain. And that's, I think, probably because that's where all the 5-HT2A receptors are. The receptors are mostly concentrated in the cortex. There's not many in the limbic bits of the brain, in the striatum and the cerebellum. But we've just reanalyzed some data, or Natalie did anyway, with a couple of students as well. We've just reanalyzed some old LSD and MDMA resting state data, specifically trying to look at these subcortical regions, because these are very important, you know, for processes like reward and emotion, and that kind of thing. So you'd think that they might be affected by these drugs, and we found that they are. In fact, we get, you know, some differences between LSD and MDMA. So with LSD, those areas tend to have more connectivity with the visual cortex, which you might expect because LSD is a classic psychedelic, and you get these very strong visual kind of hallucinatory effects with LSD. And we found that MDMA tends to turn off connectivity between those areas and the amygdala again, which we think might be why it's helpful for people with PTSD. It does tend to dampen amygdala responses, particularly related to kind of fear and anxiety and things like that. So yeah, we are starting to look at the subcortical bits, but I think most people have focused on the cortex because that tends to be where where all the 5-HT2A receptors are. So we think that's where the drug is having its kind of direct effect.
Okay. So, for example, in depression, it's primarily, well, that's the current best hypothesis, that the primary therapeutic effects are happening within the cortex.
Yes.
And not at the and not in the striatum, for example.
You know, well, I mean, people tend to associate striatum effects with disorders like addiction, because striatum is very dopaminergic, and schizophrenia, right? And whether that's true or not, I think these are all these are all kind of simplifications that we tell ourselves, you know. These are all a bit like, you know, the things we teach kids about, for example, atomic structure, right? We teach kids that an atom, it has a nucleus and the electrons spin around it, right? That's not true, right? That's basically a lie, right? It's so simplified that it becomes basically a lie, right? Atoms don't work like that, right? We know that, but it's a useful model, of a way of teaching about this stuff, right? And I think there's a danger here that we know so little about the complexities of this stuff, and there's a danger here of saying, okay, addiction and schizophrenia, they're striatal disorders, and depression, that's a more cortical disorder, when it's, you know, it's just not going to be like that. We know that the brain operates in these large networks and on multiple different levels of communication. It's, you know, it just has to be more complex than that. But we, unfortunately, unlike atomic structure, we don't even have much of an idea of what kind of complexities there are.
I see. Yeah. So you mentioned, in regards to the effects of psilocybin, that it has an effect on emotional responsiveness, and that effect might be related to amygdala activity. Can you go into that, and like, is it during the acute dosing session? Is it persistent?
Yeah. So, I mean, there's a really nice paper by a woman called Rosalind Watts, who was the lead clinical psychologist on both the clinical trials that we did at Imperial. And she did a whole bunch of interviews with the patients after the trial, and got them to talk about their experiences and how they felt. And the two themes, then she did some kind of qualitative analysis of these interviews. And the two themes that really emerged from those interviews were about acceptance and connectedness. So particularly connectedness, people seemed to characterize their depression, at least partly, as being a kind of sense of disconnection. So a sense of not being able to connect with other people, with the world, even with their own emotions, right? And then after the psychedelic therapy, I mean, one of the more kind of mystical experiences that people often report with, you know, moderate to high doses of psychedelics is this sense of connectedness to the universe, if you like, or the world, or the sense that we are all one, and that kind of thing, right? And people often reported after the therapy that they maintained this sense of connection, and that their sense of openness and connectivity to other people, and even to kind of their own emotions, they felt in touch with their own emotions again, like they hadn't been before. And I think that's perhaps related to those changes in amygdala function. So again, we saw, you know, just showing people emotional faces in the scanner, we saw this increase in amygdala responses. So I think those changes in emotional function are probably fairly important in the therapeutic effect as well.
Yeah, I see. Okay. Are there any other brain areas that we haven't mentioned that
Well,
you
I
think
mean,
are relevant?
there is this distinction. Again, it's quite general, but there's this distinction that people have developed as a way to characterize the brain, between what people call unimodal and transmodal brain areas. So the unimodal brain areas are the areas that have a specific function. So, you know, the visual bits of the brain at the back, the motor bits, the auditory bits, the bits that are doing the basic stuff: vision, sound perception, motor functions, somatosensory, touch perception, that kind of stuff. There are bits of the brain that are specialized for those low level functions. And then there are other bits of the brain, particularly the frontal lobe, which don't seem to be specified, don't seem to be specific to those particular functions. And they're doing something a bit more higher level and a bit more abstract. They're doing, you know, high level cognition or abstract thought or whatever you want to, or they're taking information from all of the lower level areas and integrating it and things like that. So the lower level areas we call unimodal, and the higher level areas we call transmodal, because they're not tied to any particular sensory modality, right? And what happens a bit, you know, as you might expect, based on what we know about what it does to resting state networks, what happens when you give people psychedelics is that this gradient between unimodal and transmodal areas breaks down. So the unimodal areas become more transmodal, and the transmodal areas become more unimodal in their pattern of responses. So that's another way of thinking about or looking at the effects that psychedelics have on the brain. And interestingly, actually, if you look at maps of where the 5-HT2A receptors are, they tend to be most clustered in the transmodal areas, in the higher level integration type areas. And we think that's why psychedelics have this very powerful and strong effect on our ways of thinking and our consciousness, because they are specifically hitting the areas that are related to our ways of thinking and our consciousness, and that integrate information from all the low level areas. They're messing up all the high level stuff, and that allows the low level areas to have maybe a stronger influence. And that's how they produce their very strong effects on consciousness and things like that.
Right. So you have to consider the the whole brain pretty much when you are thinking about a specific, well, I guess you could analyze and measure effects within a specific brain area, but in most instances, and in particular as regards cortical versus, for example, striatal effects, it's essential pretty much to analyze both and consider both when interpreting the findings.
Yeah, I mean, what I'd like to do, I mean, we've got a long way actually in figuring this stuff out using resting state data and looking at these quite general global effects on brain function. And what we haven't done a lot of until this point is to look at specific brain areas and targeting them with specific tasks. So I think there's still quite a lot to do there. I'd like to do some different emotional tasks. I'd like to get people watching movies in the scanner while we're giving them psychedelics. I'd like to do more music stuff. I'd like to do various kinds of cognitive tasks, see if we can figure out, because that's how you really start looking at specific effects and really trying to carve things up into smaller manageable chunks about how these things might be having their effects. Again, you know, it's just time and data availability and resources. But yeah, I'd like to see more use of specific tasks now. I think we haven't gone as far as we can with resting state data, but we've got a long way with the resting state data. I think it's time to start focusing on some more specific things.
Yeah, interesting. Are there any particularly important differences, do you think, as regards the effects of different classic psychedelics on either whole brain connectivity or modular connectivity, or of specific brain areas?
That's a good question. Well, with the caveat that, again, we don't have the right data, and
Course,
I don't think we have any study where they've scanned. Ideally, you'd want the same group of people and then do multiple sessions with several different drugs on different days, and then you could directly compare them. And nobody's done that. But I mean, broadly, I think they're pretty similar. So there's a friend and colleague of mine called Chris Timmerman, who's done really important work with DMT. DMT is a very, very powerful but short-acting psychedelic, so it only lasts maybe 10 minutes. And he's shown some pretty similar effects with that drug as you get with the longer-lasting ones like LSD and psilocybin. You know, there may be differences in these things, but until we actually get a study where we scan the same group of people repeatedly with different drugs, we're not going to be able to find those small differences. But broadly, they all seem to have fairly similar effects in that they produce this kind of randomization or destabilization of the brain networks.
I see. What are the long-term effects? And I'm thinking now of the effects over the course of months, for example, of classic psychedelics on personality factors.
Yeah, good question. So there's been some work looking at particular personality factors. So there's a theory of personality called the Big Five. And one of the 5 factors in that is this factor called openness to experience. And there's some evidence that psychedelics increase levels of openness to experience. There's some evidence. I mean, this is all quite early and not great evidence. There's some evidence that psychedelics increase relationships, people's positive relationships with nature and the natural world, and things like that, and make people want to go out and spend more time in nature. But really, that's about all we've got, I think, at the moment in terms of studies and data on that kind of thing. And you don't expect particularly to show big changes in personality, because I mean, personality is by its nature is something that you don't expect to change that much, right? Yeah,
that's pretty much the definition of personality. Yeah, exactly.
So the fact that we do see these slight shifts, I think, is interesting, but you don't expect to suddenly change somebody's entire personality, because that's just not how personality works.
Yeah. Yes, it's fascinating that it at all has effects on personality in the long term, I think. How would you hope that your research would impact society or treatment schedules, like how treatment with psychedelics are done?
Well, I mean, I just want these treatments to be available for patients, really. So there's only really two places in the world now. There's Switzerland, which has had these treatments available for some time, but only in a kind of limited way for kind of compassionate use, where patients really need them. And now Australia. So since last year, psilocybin has been legal in Australia for depression, and MDMA has been legal for PTSD. Again, it's still very limited use, and basically after every other treatment has failed. But I would just like to see more availability for these treatments for patients, and I hope that with Australia now we can actually start to get some studies done in Australia and show some kind of real world effects of these treatments on patients that have gone through the process in Australia, which I think is a really good, powerful set of evidence as well. You know, there's studies that happen with, you know, standard drugs, with new drugs, before they're licensed and before they come to market. And then there's often what people call phase 4 studies, which happen after a drug is available and being prescribed, showing the kind of the effects on patients in the real world when patients are actually being treated with these things. And I think if we can start to gather some good data from places like Australia where they are available, in that sense, I think that will help make the case for these things in the US and in the rest of Europe, and alongside the more traditional kind of clinical trial evidence that you get from, you know, a nice placebo-controlled trial or whatever. I think that real-world data will hopefully be quite powerful.
If listeners take away just one message from this conversation, what would you like that to be?
Oh, well, I think it's that psychedelic therapies are on their way in some form. You know, I would like, in 20 years' time, I would like to see a psychedelic treatment room in you know, most hospitals around the world. And I think it's going to help a lot of people, but it's we've still got a long journey to get there. And I'm just trying to do what I can to help us get there. That was very weak at the end there.
Yeah, I like it though. I like it. All right. Where can people learn more about what you're doing, your research?
Oh, if you just Google me, you can go. If you Google Matt Wall fMRI or Matt Wall Psychedelics, I come up. I'm pretty active on LinkedIn and Blue Sky. Not so much active on Twitter since it got taken over by Nazis, but that's fine. I'm at various conferences. There's a massive psychedelic conference coming up this year in June in Denver, in Colorado, called Psychedelic Science. If anybody's interested in psychedelics, that is the place to go. I think there's, you know, 10,000 or 20,000 people going there, from all kinds of, you know, different research areas and other things in psychedelics. Yeah, I think that's the best options.
Yeah. All right. Thank you, Matthew, for coming on the Exploring Health and Medicine podcast.
Thank you, David. It was really good. Thanks for inviting me.